Hello, everybody. My name is Ana Maldonado-Contreras. I'm an assistant professor at the University of Massachusetts Medical School. And today I'm going to talk to you about a diet that we created here at school in conjunction with the Center for Applied Nutrition, which is led by Barbara Oldansky. And the title of the talk is IBD Anti-inflammatory Diet, or IBD-AID. And it's the proof of concept study that I'm going to share the results with you. So before we start, I wanted to talk a little bit about the pathogenesis of inflammatory bowel disease. It is thought that there are four main factors that contribute to the onset of disease. So this disease happens in hosts that are genetically susceptible for the disease. There have been around more than 300 loci in the gene that have been associated with increased risk of developing inflammatory bowel disease. Most of those genes are related to how our gut interact with the environment, and in this case, with the microbiome. So these host genetics of these genes can be regulating or promoting a dysregulated immune response to commensal bacteria that normally habitate our gut, or also these host genetic loci that have been linked to risk to IBD can promote a barrier dysfunction, which allow the microbiome or bacteria components of the microbiome to breach the epithelial barrier and then to get into the luminal side and promote a very strong inflammatory response. However, not only host genetics are explain the risk of the disease, there seems to be an environmental trigger that also play a critical role in the onset of disease. And we are interested in the role of the microbiota, these millions of bacteria that live in our gut that can also, depending on the population, can promote a dysregulated immune response or can promote barrier dysfunction. The microbiota can be regulated by several factors, and one of the main regulators of the microbiota are driven by diet. So we have hypothesized that diet can actually feed the microbiota in terms of favoring bacteria that are beneficial from in terms of promoting a regulatory immune response, regulate inflammation or lower inflammation, and strengthen epithelial barrier in patients with IBD. So with that will improve the health of the patients suffering from the disease. So we designed it, as I say before, a diet. It's called the IBD anti-inflammatory diet, and I'm taking a lot of credit for it, but the person that led this effort was Barbara Oldansky, as I say, for the Center of Applied Nutrition. And she and her team of registered dietitians come up with a diet that is mainly supported by four components: probiotics food on beneficial foods, and exclusion of foods that are known to trigger GI symptoms or intestinal symptoms and dysbiosis. So prebiotics foods are those that are used by commensal bacteria, and we expect that by providing these probiotics foods, we can increase the commensal and the beneficial bacteria in the guts. And some of the examples here are oats, legumes, peas, nuts, mostly vegetables and fruits, high in fiber foods. Then we have probiotic foods that are all mostly fermented food, like yogurt, miso, kefir, sauerkraut, to also incorporate beneficial bacteria that are already in those foods, and then increase the diversity of the microbiota in the gut. Then we also need beneficial foods to support nutrition, and we encourage with the IBD-AID the increased consumption of beneficial fatty acids, such as mono and omega-3 fatty acids, and lean proteins and healthy fats. Among the foods that are excluded in the diet, we have sucrose, wheat, corn, potatoes, lactose, trans fatty acid, and saturated fats should be limited. So there is more information, and we have compiled more than 500 recipes on our website. Here you can see it. And you can go look, these are just only examples of some of the foods that are included in the IBD-A and the ones that are excluded. And we also have, besides having like a patient curriculum where with all these recipes, we take the time to bring the patients to the kitchen to teach them how to cook. And here I'm showing you a master class of some of the recipes, IBD approved recipes with some of our patients and some of our actually medical students. We also teach them how to cook IBD-A, so they can, when they get to their clinical work, they can actually answer the most frequent question asked for patients with IBD, which is, what should I eat? And as I said, we also have very in-depth website with different recipes per phase, for breakfast, for entries, and so patient can actually access the foods and the recipes online. So we started to have, this diet was designed to revert dysbiosis. So these patients and our patient population, and worldwide patient population with IBD have this dysbiotic microbiota, which means there are less of the beneficial bacteria and more of the pathogenic bacteria. Then that triggers a dysregulation of the immune response and an exacerbated immune response, and also lower the epithelial barrier function, creating this leaky gut. So can we feed the good bacteria and the microbiota? And that was the whole concept behind the diet. Which foods can we use to strengthen the microbiota, the good and beneficial microbes in our gut? So we did started a proof of concept in 2017, and we have 4 questions. Can patients achieve dietary compliance, which is crucial for dietary therapy? Those adopting this diet, the IBD-A, reverts dysbiosis, this again, altered microbiota seen in patients with IBD. Also, we were interested in knowing which foods are related to the changes on the microbiota, so we can further personalize the diet that can be customized for patients. And also, which foods are the foods that we are trying to introduce in this diet? Can those foods modulate the immune tone? So to answer all those questions, as I say, we started this proof of concept study from 2017, and we finished in 2018. Here is just a flow chart of what our recruitment looks like. We recruited 25 patients. 3 withdrew. We did a baseline of 22 patients that then ended up, some of them withdrawing, and we ended up with only 19 patients completing the diet. So we have, we obtained it. Here is on the right is the study design. We have a baseline. So we use the patients as their own control, given that the microbiota is very personalized and it's very different person to person. Also, the response to diet can be very unique per person. So we use the patients as their own control. We did a baseline to determine like baseline levels of dysbiosis of cytokines, and also dietary intakes. So that was for 6 weeks. We took dietary information. We took collect stool samples 2 times per week to analyze the microbiota, and we also took blood once at baseline. And then the intervention started, and it was for 8 weeks, and we did exactly the same measurements to compare before and after the diet. In total, we got more than 500 24-hour food queries. That's how we assessed the dietary intervention or what the patients were eating. So we have around 29 of these questionnaires per person. We collected over 300 stool samples, around 17 samples per person. And we collected only 18 blood samples. Only 9 patients out of the 19 were able to give us before and after the diet blood samples. And I'm going to show you some of the results. So the first question we have was, can patients achieve dietary compliance? And we saw a dramatic change in their diet and patients' diet before and after intervention. So I'm here showing you some of the 4 arms of the diet. As I say before, prebiotics and probiotics, beneficial foods and adverse foods. Here on the x-axis, we can see the components of that food category. Like for example, in prebiotics, we have fruits, vegetables, oats, and honey. And we can see in red where the average intake of prebiotics at baseline, or in blue, the average intake of prebiotics at intervention. Again, and the asterisk means significant. So we saw a significant shift on the diet in patients that went through the intervention. It was significant for all the foods included in the prebiotic category. It was significant only for the fermented dairy foods, yogurts mainly for probiotics. And for the beneficial foods, we saw a significantly increase of intakes of fatty acids, healthy fatty acids, omega-3 and monounsaturated fatty acids. And then adverse food, and not surprisingly, it was the most significant on all the foods that were included in that category. It's easier for the patient to avoid foods that are known to be triggering, that actually add other foods for fear of flare or we're eating those new nutrients. So again, we see a decrease of wheat, lactose, high-fat animal protein, all the foods that are that we group into the adverse group. Not only patients changed their diet, but the diet was adopted very fast. Here I'm showing you those foods that were significantly different between baseline and intervention. And here in the circles are the mean serving sizes that patients reported at baseline in red, or during the intervention in green, in blue. And as you can see here, the beneficial foods such as prebiotics or omega-3 fatty acids, there is an increase. You can see the blue circles are bigger on the intervention, meaning that the serving size increased during the intervention, while the adverse food, all the circles are small. And the differences start right after the first week of intervention here at week 7 and 8. So patients can change their diet. These participants can change their diet, and it can be a rapid change of the diet. So there is compliance to the diet. The second question we have is, does adopting this IBD-AID reverse dysbiosis or change the microbiota to a beneficial one? And we saw also that along with the intervention, there was a drastic microbiome change after the IBD-AID intervention. So here I'm showing you in the top panel the results for all the participants together, the 19 participants we have. Here, each of these bacteria and the bar represents how much enrichment of that bacteria at the intervention in green or at baseline. So for all the participants, the top bacteria that increased at intervention was Roseburia hominis. And then all these are significant too, but the top one seems to be Roseburia hominis. And then enriched at baseline, we have a bacterium, Collinsella, another bacteria that there are normal commensals. Some of them are being related to inflammation, but here the bacteria that are enriched at the intervention are the ones that has been non-depleted for patients with IBD, and also known to produce short-chain fatty acids such as butyrate that also maintains the gut homeostasis. So then if we break it down by Crohn's disease of ulcerative colitis, we see differences in the microbiome shift during the intervention. In the CD patients, Roseburia hominis is still being the top one. First bacteria that seems to be enriched after the intervention. And then we have for ulcerative colitis, Faecalibacterium proudnici. All of these bacteria that I'm showing you here that are enriched in intervention are like the hallmark of this biosis of alteration of the microbiota. In all the studies worldwide, in worldwide cohort has shown depletion of these bacteria in patients with IBD. And after 8 weeks of the intervention, even during the intervention, these samples are taken during the intervention, we start seeing this dramatic shift favoring bacteria that are usually depleted in patients with inflammatory bowel disease. In the case of baseline, one of the things that we are attentive to is there is this Prebiotics dystazonis that seems to be relevant or enriched in baseline in both in patients with Crohn's disease and patients with ulcerative colitis. And we are looking into the role of this bacterium in inflammation in the context of inflammatory bowel disease. Then we also saw an enrichment of the microbial genes that are dedicated for butyrate and acetate production. As I say before, butyrate and also acetate are short-chain fatty acids that are generated by the fermentation of fiber by microbes. And as these bacteria, the slide before I show how these bacteria increased, all those bacteria have a genetic capacity for butyrate. So we'll look at the genes to see if we also see an enrichment of the genes in the microbes that can produce this short-chain fatty acid, and indeed we do. And it's exactly the same picture or graph that I'm showing you before. This means that are enriched in intervention. All these bacteria, all these genes that are associated with butyrate or acetate production down here are enriched after the IBD. So we have a chief of the bacteria in microbiota composition that is related to increase of butyrate healthy commensal bacteria that are depleting in IBD, and along with that, the butyrate production and acetate production genes are also enriched after the intervention. So then we were interested in knowing which foods are related to changes to the microbiota, and we did studies of correlation with bacteria correlate with the most changes on the microbiota. And here I'm showing you the top bacteria that were enriched in baseline for patients with Crohn's disease here in red, or the top bacteria that were enriched and during the intervention in blue. And here in the bottom, I'm showing you the foods that we found correlation with. In blue are in circle blue with the circles with blue are a positive correlation. So we see all these bacteria that are enriched in baseline are positively correlated with consumption of adverse foods, while the ones that are enriched during the intervention are positively correlated with prebiotics and with mostly prebiotics and the prebiotic foods included in this food category. We have negative correlation of Firmicutes that was enriched in base intervention with fried vegetable, meaning that if you increase the consumption of adverse foods, you will have, the correlation is that you have higher abundance of these bacteria during baseline. Well, when we change and we include more prebiotics in our diet, an increase on prebiotic intakes is correlated with a higher abundance of these bacteria that, as I say before, are butyrate and acetate producers. Same story with UC patients. In red, the ones that are associated or enriched at baseline, and in blue, the ones that are enriched during the intervention. Here we see this Faecalibacterium proudnici, which is again a signature bacteria that is depleted and missing in patients with UC, and you see that there is a negative correlation, meaning that as much by eating more adverse food, we decrease the abundance of these bacteria in the gut. But then other bacteria that are associated or enriched during the intervention are positively correlated with beneficial foods, with prebiotics and with probiotics. So then the last question for us was to was to look at the foods in IBD-AID, in our IBD-AID diet, and look at if they can modulate the immune tone. So we collected, as I said before, 9 patients give us samples before and after diet, and we measured this 14 cytokines that are related to inflammation in IBD, and then correlate the levels of those cytokines with all the food components that were reported by the patient. And what we saw is that prebiotics and beneficial foods, which both are encouraged by the IBD-AID, correlate with lower levels of pro-inflammatory cytokines, even at baseline or at intervention. So these 9 patients, subset of 9 patients that we have, the ones that have higher levels or higher intakes of prebiotics were the ones that have lower levels of IL-6 and IL-8. Both pro-inflammatory bacteria, both increase in patients with IBD in having an essential role in perpetuation of inflammation in these patients. And on the contrary, we saw that the adverse foods correlate with higher levels of pro-inflammatory cytokines. So here we did exactly the same analysis, and we saw that of these 9 patients, the one that ate more adverse food have the higher levels of pro-inflammatory cytokines. And here we're talking about IL-8 and TNF-alpha at baseline, at intervention. And we see, for example, here, foods and beverage high in sugar were very well correlated with higher TNF-alpha at baseline. So to summarize this, we're seeing that the foods that we're including as to increase during the IBD diet can have an immunomodulatory tone or role in IBD. So to conclude of this proof of concept study, we show that patients can rapidly adopt the IBD-AID, that the IBD-AID reverts dysbiosis and is independent of IBD subtype. It happened in both Crohn's disease patient and ulcerative colitis patients. We found that prebiotics, probiotics, and healthy fats seem to be the ones that are key to increase butyrate producing species in these patients. We saw that prebiotics, specifically fruits, are correlated with lower levels of pro-inflammatory cytokines as interleukin-6 and 8. And adverse foods were correlated with higher levels of pro-inflammatory cytokines like IL-8, interleukin-8, and TNF-alpha. So these have major implications, and we believe that this is a proof of concept of a dietary intervention targeting the microbiota that can be used as an adjunctive therapy to treat patients with inflammatory bowel disease. You have to stop.